I'm very pleased to present EFSTAR here at this conference, and the title of my talk is how EFSTAR is approaching this challenge of developing bispecific antibodies. We've already heard about the manufacturing challenges and developability challenges for monoclonal antibodies, and this is something that really extrapolates and is magnified with bispecific antibodies. But first, maybe let's step back why an interest in bispecific antibodies altogether. And I'm going to show a few slides from a study published by Jan Eidek, where they worked on a bispecific targeting EGF receptor and IGF receptor at the same time. And as you can see here, the scaffold is a very classical way of making bispecific antibodies, where you bolt on either the N-terminus or the C-terminus of the antibody. You bolt on an additional binding domain, a single-chain fusion protein that is targeting a second antigen. And as you may imagine, the challenges that David talked about earlier about predicting developability is a huge challenge with this kind of format, because you have a secondary structure that is not native to the Ig molecule from its evolution, and can create separate aggregation problems and challenges as you progress through development. But basically, when you look at the results that was obtained in this study, and it's really one of the only studies where you have seen a rigorous comparison between bispecifics and the constituent monoclonal antibodies. You see here what is the aim. Basically, the aim is to achieve a synergy between the two binding domains. So you have EGF receptor and IGF receptor in either configuration, and then you have a synergy here, and here you have the constituent monoclonal antibodies as combination in a mouse tumor xenograft. And basically what you see is that this bispecific provides an additional biology and additional synergy compared with the combination. You also see that if you flip the orientation around, and you can do this in multiple ways, you could, for example, switch these two or put one up here and so forth. There's multiple permutations. You see that it's not interchangeable. You don't necessarily predict your biology simply by having two good antibodies and then you put them together. There's a lot of experimentation required to establish what is the right structure of your bispecific antibody in this type of approach, and also what is the right combination. But ultimately, the aim why bispecific is to achieve this kind of improvement in your response over the combination of the two antibodies. What are the main issues with bispecific antibodies? I think the greatest challenge is to actually find the right combination. So if you have two antibodies, how do you actually identify the two partners for your bispecific that will actually give you that synergy? And quite honestly, it's not possible to predict it. You really need to do it experimentally. It's an iterative or deductive process where you need to look at many combinations. And that's, of course, a challenge if you have to spend a lot of engineering time to prepare your bispecific antibody. And then subsequently, once you've actually developed your bispecific, once you've discovered, your bispecific, as what David was talking about, then you have all the druggability issues. So is it manufacturable? Can you scale the manufacturing even when you see good manufacturability at small scale? What's stability and PK and issues of immunogenicity? These are really the key aspects of bispecific challenges. The main goal, of course, is that you have additional biology that is not achieved necessarily by the combination, so the efficacy improvement or the synergy. This can be achieved in different ways. So cross-linking of receptors, you can have increased downregulation of your targets, you can have increased local concentrations of your one antibody due to tissue targeting through the second antibody. So there's different ways, theoretically, you can have improvements or synergies obtained in a bispecific approach. But probably key, you need to have a biological hypothesis, and then you need to be able to test that. And in EFSTAR, our approach is really to use the genuine IgG format to create bispecifics, and I'll explain to you why that's important in our view. Basically, we have a highly modular platform. I'll come back to that. We have two validating partnerships, one with Merck Serono and one with Boehringer Ingelheim, where we discover on a target-by-target basis compounds for our partner with a good, strong IP portfolio. And we are now, all of the company is now located here in fact, here on the campus in Babraham. And I joined the company last year and is very proud to work with the team and and with our exciting technology here in Cambridge. So what is the approach that we use to develop the bispecific antibodies? At the heart of the technology, we have this scaffold or this molecule that we call the FCAP, or an Fc domain with antigen binding characteristics. So basically what we do is we take the Fc domain of a classical IgG scaffold. So what we use is the IgG1 human sequence. We create libraries, and then we select for antigen binding in the end of the CH3 domain down here. And basically what this then generates is the normally non-antigen binding part of the antibody. We develop a fragment of that which will bind to antigen and basically constitute a module that you can use to create a bispecific antibody. In principle, this can have the similar potency as a normal antibody. So for example, due to the localization here, there's no impact on, in general on the Fc receptor binding, and the effector functions are retained. Also, the half-life, if you inject this as a molecule itself in mice, is 75 hours approximately, which is much greater than many of the smaller scaffolds. So it could be considered a drug in its own right. The way we do it is, so again here a blow up of the CH3 domain and the CH2 domain. We focus on this part of the CH3 domain down here. We variate the sequences in the A, B, C, D, and EF loop in the CH3 domain and of the wild type FC region, IgG1. And then we use phage display and yeast display to basically select binders out of libraries that have diversity in these regions. Typically we have in the range of 10 to 12 amino acids that are changed away from their wild type sequence in the final products. The initial output of the selection is typically of low affinity, and then subsequently we go through repeated affinity maturations to generate high affinity binders to the antigen in question. Diagram showing the timeline, basically 6 to 10 months to get to the first hits out of the selections, and then a period of perhaps a little bit variable length subsequently to go through the subsequent affinity maturations to get typically low single-digit nanomolar affinities. Then the opportunity that this presents is that once you have the antigen-specific FCAP, then you can simply, in a very modular manner, slot that module into any IgG molecule. And you can do this almost in high throughput. You can do it early in your discovery. You can do it before you're really locked into your particular target combination. You can have several FCAPs. You can have several FAPs, either against the same antigen or against multiple different antigens within a certain therapeutic biology that you would like to interrogate. So you can very quickly and modularly establish in your early discovery, so push back basically the engineering question, push that back to your discovery and say, which biology can we generate by testing a number of different combinations. And then at the end of the day, you have a bispecific that really looks like a monoclonal antibody, retains all the normal functionality of monoclonal antibodies, and predictably will maintain the normal monoclonal antibody manufacturing characteristics. So we're not adding any additional manufacturing liabilities, which is really the case with almost all other bispecific scaffolds, that they don't really predictably manufacture. And then hopefully through the biology, through your discovery of the right combination, it's possible through the modularity of the platform, it's possible to identify combinations that result in a strong synergy of your final product. So here a slide really sort of illustrating perhaps very simply this issue of the modularity. You can take the FCAP and really quite easily put it into any biospecific combination and use that in your early testing. And it's not, I want to make sure that this is clear, it's not a trivial issue with normal bispecific antibody development, because as you can imagine, if you have a very challenging scaffold, like a single chain domain fusion protein, where you need to work on the linker length, you need to work on amino acid replacements to ensure you have stability and so forth, you invest a lot of energy and time and money in your single molecule, and this is something where, in a modular manner, you can actually wait until you have your final product in terms of the biology combination, and then check on your developability, which is an important benefit. So, need for sophisticated biology-based screening. This is what the modularity allows in your early discovery. Not just any combinations of targets will result in your synergy, so you need to be able to build that into the approach. And then the favorable CMC properties due to the similarity with the IgG molecule itself. Just a few words towards the end about our lead program. We have focused on a particular range of targets, the B family of antigens within oncology. We have a biological focus in oncology in the company. The antigen HER2 will be familiar to many of you as the target of approved drugs, trastuzumab, or also called Herceptin, and pertuzumab in combination with trastuzumab in certain indications, also called Perjeta. And in this space, we believe that there is, although it's of course a very competitive area, we believe that there is an opportunity to improve on standard of care for non-responsive patients in this area. And we've identified a very potent HER2-specific FCAP, which binds to a different epitope than Herceptin, and displays some very promising biology in our early in vivo testing. The FCAP will inhibit proliferation and induce downregulation of the receptor and inhibit the phosphorylation of receptors, similar to some of the other agents in the space, and in that case behaves as you would predict for a compound against the target. Very briefly about the model that we are using to explore the in vivo biology. It's a model which is called the Oncotest in vivo xenograft tumor models. Basically what the company Oncotest is doing for us is taking primary tumor explants. So these are tumor explants that have not been propagated in tissue culture, except for just the very brief establishment of the tumor in the mice during the experiment. And then you use for the propagation, you use immunodeficient mice to basically establish these human patient-like tumors from individual patients in each of the experiments. And then you can use your test compound to test whether the test compound will actually inhibit the tumor growth in these models. I'm just going to show you, I think, one slide here showing the performance of the HER2 FCAP. So this is not the bispecific yet. This is the data from the initial testing of the HER2 FCAP itself. And what we've seen in some of the models, not in all of them, but in some of the models, we've seen this extremely potent phenotype or extremely sensitive phenotype of the tumor models. Basically, the FCAP upon 5 weekly single treatments at 10 mg per kilo, the FCAP completely eradicates the tumor and there's no regrowth. And we're trying to tease out what is in fact the phenotype of these tumor models, and we have a patient stratification or a patient tailoring a biomarker theory about why that is that these tumors respond like this, which in fact we have used to then select tumors and shown that there's a predictability of this particular phenotype that we have identified, and we're using that as part of our development strategy going forward. Some of the mechanisms of actions I already mentioned, that downregulation of HER2, inhibition of HER2-mediated functions either through dimerization or inhibition of dimerization, blocking phosphorylation of downstream pathway elements, or antiproliferative activities. Also apoptosis and ADCC could be putative mechanisms. But where we are at the moment is basically taking this FCAP, we're testing the FCAP itself in the context of a number of different monoclonal antibodies that we have combined this into. And the key take-home from this slide really is perhaps the modularity and the speed in which we can do this. So imagine with another bispecific antibody technology, you go from inception. Basically, you have your combinatorial agent at this time point, and then 2 quarters later, you have your in vivo data. There's not really any other platform that will allow you to do that with 4 different bispecific antibodies derived from a theoretically predicted good combination within that short time span. So this modularity is a really powerful aspect of our approach. And if you look at the challenges with bispecifics, that's one of the key challenges. So how will we differentiate this program? I think this is almost my last slide. How will we differentiate the program? Basically, we believe that the ERB receptors as an attractive target within oncology. We believe that within the HER2 space that there is a definite room for improvement. Herceptin itself has very limited single agent activity and a high proportion of patients are non-responsive. The combination of Herceptin and pertuzumab have improved, but still, really, you don't really see any cures at any great degree. Then there's a TDM1 product, which is an antibody-drug conjugate version. Also has certain benefits, but the side effect profile, we believe, will potentially limit the use of that compound. So our aim is to use the bispecific antibody and use potentially the ability to block multiple pathways or have a stronger blocker of an individual pathway to generate synergy and improved clinical potency. And then we have, as I mentioned, a patient biomarker for predictive response biomarker that we believe will identify those subcategories of patients that shows this very strong sensitivity to the agent, and in that we hopefully will be able to generate improved overall survival. And with that, I'd like to thank again for your attention and thank you to all the F-Star members of the team that are present here. I think we have 15 people here today. So I'm not going to show names of, it's really the whole company that has generated this fantastic platform. So thank you.